morning, everyone, and thank you very much. As a Bostonian, I too would be home shoveling my driveway, but unlike New Yorkers, I have the memories of World Series victory to keep me warm. I think 4 World Series victories.
27.
It's called winning over the crowd. I guess I did a good job. So, I'm at Dana-Farber Cancer Institute in Boston. A large portion of my job is actually treating lymphoma and running clinical trials in lymphoma, but I also have a history and an interest in histiocyte disorders as well. So, this morning I'm going to give an overview of the clinical trials process, sort of the sausage making and how we think about trials, how we conduct trials. It's not necessarily a lot, to specific Erdheim-Chester disease trials, but I'm certainly happy to answer any questions in that regard. And the talk is not meant to be formal, so please interrupt at any time with questions. Booze if I mention the Red Sox again. Nor is it comprehensive. So for those of you who are intimately familiar with the process of bringing drugs to market, you'll note that I did skip some important things. So when I approach patients about clinical trials, they have a lot of questions about exactly what it is that we're studying. What are these different types of drugs? And historically, oncology has really utilized chemotherapy, which are drugs that target important processes in DNA replication, cell division, cellular metabolism. So they're relatively non-specific drugs, and therefore, many of them have fairly significant side effects: the nausea, hair loss, low blood counts that we often see with chemotherapy, because these are drugs that just essentially target rapidly dividing cells. They're not very specific. The field has really steadily moved away from chemotherapy. We don't see a lot of chemotherapy trials anymore, at least trials of new chemotherapy drugs. And we've really moved into the realm of immunotherapy and targeted therapies. So immunotherapy refers to a broad class of agents that somehow utilize the immune system to attack a disease or attack a cancer. And there's a variety of ways of doing this. Probably the most common class of immunotherapies are antibodies, so proteins that are engineered to restore or help the body's ability to attack a cancer. Vaccines are being developed in a variety of different cancers. I have particular interest in lymphoma vaccines, although to date, none of them have really been broadly applicable in the treatment of cancer. And then a new technology that you might have read about called CAR T cells. It's really a mechanism where we genetically reprogram a patient's immune cells to attack and destroy the disease. So it's very exciting technology, and we're really at the dawn of that technology. Relevant to Erdheim-Chester disease are what we call targeted therapies. So again, this is a broad term, but targeted therapies, as the name implies, are much more specific than chemotherapy. So they generally target a cell signaling process that's important in the growth and the development of the disease. So it's sort of a smart bomb that hones in on the target rather than carpet bombing with chemotherapy. And again, there are a variety of these that are used and in development. Relevant ones for this discussion are vemurafenib and cobimetinib and other drugs in those classes that are used to treat Erdheim-Chester disease. Then we get lots of questions about gene therapy. Gene therapy is really in its nascency. People have been at it for a long time. To my knowledge, there's only one FDA approved gene therapy at the moment, but it is a rapidly emerging field. You may have heard of this new technology called CRISPR, which is a way to individually edit genes, and it's a technology that many people think will rapidly expand this field. But for purposes of Erdheim-Chester disease and many of the malignant conditions that we see, most trials nowadays revolve around either immunotherapy or targeted therapy. What are the benefits to the patient, and what are the benefits to us to even conducting clinical trials? Well, for patients, trials offer an ability to access new medications. So many of the drugs that we study are not available outside of a clinical trial. So it sort of expands treatment options, gives patients additional options. And And there have been some studies to show that patients actually have better outcomes when they're treated on a clinical trial versus when they're treated with standard of care regimens. There's a variety of reasons why that may be true. I think the probable reason is when you're on a clinical trial, you often have a research nurse. Speaking of our own experience, and I'm sure at other institutions, research nurses are fantastic. They're patient advocates. They're closely monitoring your symptoms, your side effects. It's really sort of an extra buddy to help you through the treatment process, and they're very good at detecting and managing side effects. On a global level, of course, trials improve our scientific knowledge, and I'm always amazed about the altruism of patients. So when I approach patients about clinical trials, almost always their first question is, will this help someone else? And I find that really remarkable and uplifting, especially after watching the morning news, which is often not uplifting. So it restores your faith in humanity. Despite some of these benefits, only about 5% of adult cancer patients in this country participate in clinical trials. So the vast majority of patients never touch base with this experience. Tried to list a lot of the concerns or questions I hear from patients about participating in trials, and the number one question or concern is almost always, I don't want to be a guinea pig. And there's some concern that they may be the first person ever receiving this drug, or we have very little knowledge of it, or no idea how it works. Those are legitimate concerns. I think in many cases, as I'll show, we've done due diligence before we launch these trials to be reasonably confident that drugs will work and will not have prohibitive side effects. But we're not perfect, so there is always that element of the unknown. My kids own guinea pigs, so in addition to not wanting to be a guinea pig, I would not advocate buying any for your children either. Patients are often concerned that they're going to receive a placebo. Placebo studies are relatively uncommon in oncology, that we do use placebo on occasion. But you will always be aware if there's a possibility you're going to get a placebo. So we never do a bait and switch, where you sign up for a study intending to receive a medication, we kind of slip in a sugar pill. So you'll be notified, you'll be very aware if there's a possibility of receiving a placebo. There are legitimate travel and cost concerns for patients participating in clinical trials, particularly true with rare diseases like ECD, where there may be relatively few centers across the country that have expertise with the disease or have clinical trials studying the disease. So often travel distances can be prohibitive if you're not feeling well or if you're having side effects. It can be difficult to sit in Boston traffic or difficult to travel back and forth via airplane to a study center. And of course there are costs involved in that. There are a number of different philanthropic organizations that will often help offset some of these costs. So there are patient funds that are often available to help with travel costs that are part of clinical trials. Trials usually do involve more visits to the office, and one increasing concern from patients is trials often involve more scans, so PET scans and CT scans. And there are legitimate concerns about some of the possible adverse effects of radiation associated with diagnostic tests like CT scans and PET scans. So we're cognizant of that, and as we design trials going forward, we try to really minimize the amount of testing and the amount of scans to the absolute minimum necessary to safely conduct the trial and to get a cogent result from the trial. You do have less scheduling flexibility in the trial, so there's certain times when you have to be in clinic and we have to try to stick to that schedule versus if you're coming in for a regular office visit, you don't necessarily have to be as strict with the schedule. And patients often worry about the loss of autonomy. Gee, if I sign up for this trial, I can't back out. I'm committed to this. And I always emphasize that when you sign a consent form, it's not a legally binding contract. So you can decide to withdraw participation or withdraw consent at any time. I think patients worry that that may not be the case. But by far and away, circling back to the guinea pig issue, I think one of the biggest concerns for patients is just the unknowns about the effectiveness or possible side effects of the disease. So how do we try to alleviate some of those concerns? Well, in the modern era, when we bring a drug to a clinical trial in humans, it's generally been extensively tested in what we call preclinical models, so the proverbial petri dish. or in animal models. And one way of doing that is basically to take cells and spread them on the equivalent of a petri dish and expose them to various drugs or combinations of drugs. And you can get some sense as to whether or not the drug actually kills the cell, kills the intended target. Now that can be done very quickly and on a very large scale using automated mechanisms. So processes that used to take months or years can be sped up significantly. So at least as we go into animal testing and eventually human testing, we have a pretty good idea if the drug is going to do what we intend it to do. When you study something in a petri dish that does have limitations, you have an isolated cell, it's not being supported by other processes in the body, it's not interacting with its environment. We know that the environment of a tumor or a given disease is important in how that disease behaves. So increasingly, we're also utilizing something called xenograft models, where we can actually harvest tumors from patients and implant them in mice. Mice don't have lawyers, so we can do these types of things. And these mice are immune deficient, so their body doesn't reject the human tumor, so the human tumor grows in the mouse. And then you can essentially run clinical trials in a mouse. You can give them medications and see if these tumors shrink. There's some limitations to that. Mouse physiology is not the same as human physiology. These mice are immune deficient, so for studying immunotherapies, we might not get a good idea of how the immune system interacts with the tumor. But at least gives us a good inkling in an animal how well these drugs may work. And this is not a comprehensive list of methods of preclinical testing, but just to give you some idea of how we might approach this. And I'll put in a plug for my colleague David Weinstock, who created this company called Proxy, which now offers over 200 of these xenograft models, essentially free of charge to researchers across the world. So the only cost is the shipping cost and the cost of the mouse. So kudos to him. He made that nonprofit. So once we've made it through this preclinical or this pre-patient testing, there are a variety of phases of clinical trials. So most trials are either phase 1, phase 2, or phase 3, at least when we're talking about drug trials. Historically, the primary purpose of the phase 1 trial is to determine the safety of the drug and then to determine the optimal dose moving forward. That's still somewhat true, but again, with modern preclinical testing techniques, we have a pretty good idea if a drug may work, and so we kind of cheat. So phase 1 trials nowadays aren't necessarily solely focused on safety. We're also trying to see if there's a signal as to whether or not a drug is effective. Once a drug passes phase 1 testing, it usually moves into phase 2, which is a larger trial, often at a fixed dose, although sometimes at different doses of the drug. And it's really a better way to get an idea of how well a drug works and how safe it may be. Some drugs are approved based upon phase 2 trials, and not all drugs move to phase 3 testing. Particularly in oncology, many drugs are approved in phase 2. Phase 3 tests are generally what we call randomized trials. So patients are randomly assigned by a computer to what's considered the standard of care treatment for that disease versus the new treatment. And that's really the gold standard to prove if a new treatment is better than an existing treatment. There are other types of trials. There are phase 4 trials, which is generally post-marketing surveillance, so to look to see if drugs have unexpected side effects as they've been on the market longer. And then again, important to ECD, there are a variety of different non-treatment trials, particularly registry trials, where we collect information about patients, about their disease, about their treatments, about their quality of life, to learn more about a disease process independent of studying a new treatment for that disease. So if I'm sitting at my desk in between emails, texts, pages, cell phone calls, and people knocking on my door, what do I do if I'm thinking about starting a trial on my own? Well, the first thing is you have to find someone to support that trial. So these trials can often be very expensive, and you need some form of funding mechanism. There's a variety of sources of funding. It might be a pharmaceutical company who manufactures a drug that you're interested It could be a federal government grant, it could be a philanthropic grant, but in some way you have to identify a funding source, and then you have to often submit something called a letter of intent, which is a brief summary of what you intend to do and how you intend to do it. That's reviewed by the funding source. Sometimes they agree with and like your concept. Many times they don't, and often the process might end there. If we get preliminary approval, particularly if we have grant funding, we're generally ready to move forward. At our institution, in addition to this initial review, we have another committee that reviews your preliminary concept and adds input and strengths and weaknesses of the trial and how it may be improved. From there, we sit down and write the entire protocol. It's a document that routinely is 100+ pages, as well as the consent form. And if any of you have ever signed up for a trial, you know that these consent forms can be Byzantine and 30 pages long, 2/3 of which is written by lawyers, and then 1/3 of which kind of has some of the medical information. Once we've written all those documents, we submit them to our scientific review committee so they make sure that the trial is scientifically valid and can answer the questions it's designed to answer. And then it goes to something called the Institutional Review Board, so where nursing, pharmacy, scientists, clinicians review the trial, ensure that it's ethical, ensure that patients are adequately informed of the risks and benefits, and then again, that it can reasonably answer the question it's intended to answer. Once it passes all of these steps, the protocol is activated, so somebody has to write all the treatment plan into our electronic medical record that has to be reviewed by pharmacy and by nursing. So this process, now we're probably a year plus into the process before we've actually even intended to treat a patient. Then the trial actually opens and accrues patients, and we record the data. That requires quite a bit of staffing, so we have research coordinators that follow every patient and record all the data, side effects, effectiveness, results of tests. They have to report side effects and any issues to regulatory authorities. And then eventually the trial is published, so we have a lot of help with that from statisticians and other professionals. So you can see this is a long process, and I think sometimes patients get frustrated, why does it take so long to do all this? Well, it's a pretty Herculean effort. It requires a lot of staffing and a lot of work. Certainly well worth it, but it's not an easy or straightforward process. Once the trial is up and running, how do we determine success? Well, historically in cancer studies, success is really focused on response rates, is the tumor shrinking, and how long those remissions or those responses are lasting. And that's still important, but again, in many diseases and relevant to Erdheim-Chester disease, that's really not the only or necessarily the most important metric of success. At the end of the day, you have to realize there's human beings behind all this, and how they're feeling is really important. So there might be situations where a scan doesn't look a whole lot different, but the patient feels dramatically better. Well, 15 years ago in oncology, that wouldn't be considered a success. Now we've started to rethink that and say, okay, the patient feels better. Does it really matter as much what their scan shows? On the flip side, maybe a patient's scan looks fantastic, but they feel horrible because of side effects from the drug. How much have you really achieved in that circumstance? So I think we're not perfect, but we're doing a better job of sort of analyzing the whole person and not just how their scans and blood work look, but how they feel on these treatments. And apropos of skyrocketing medical costs, there's some studies now, particularly larger studies, look at resource utilization. How much are these therapies costing relative to the benefit that we're getting from them? How much are they costing the patient? Are we bankrupting people because their co-payments are $2,000 a month for some of these drugs? Also, in many settings historically, we've kept people on treatments indefinitely if they're not having side effects or problems from the medication. Is that always necessary? Is that always in the best interest of the patient? So these are important questions that until very recently haven't always been emphasized in oncology trials. Sort of the ultimate metric of success maybe is FDA approval. We're very fortunate to have an FDA approval in Erdheim-Chester disease. It's a testament to the people in this room, to Kathy and her organization. I think Diamond and Hirosh in the back rows there have really led that effort, and I was extremely impressed that we had an FDA approval in Erdheim-Chester disease. Historically, it takes about 12 years from the time a drug is first tested until it's FDA approved. That has shortened of late. To my knowledge, the drug with the fastest approval from first-in-human studies to FDA approval was a drug called ibrutinib that was approved in 4 years from first-in-human testing. There may be others that have been approved more quickly, but it's still a process. I've seen various estimates as to the cost. Best estimates range from $350 to $800 million. I saw one estimate of $2 billion, but that was from a pharmaceutical industry source, so they obviously have an incentive to inflate some of the costs. But I think somewhere in the $500 million range is a pretty good estimate of what it costs to bring a drug to market. So that's a big investment by any measure. On average, there's about 3.5 years of preclinical testing, and historically about 99.9% of drugs have failed at that stage. So talking, many are called, few are chosen. And then roughly, it takes 1 year for phase 1 testing, 2 years for phase 2 testing, and 3 years for phase 3 testing. So as you march through that process, the years add up. And then according to one reference, a new drug application to the FDA was 100,000 pages. Hopefully a PDF file or not printed. And the application took about 2.5 years. Now, to the FDA's credit, they've taken a number of steps and introduced a number of mechanisms to accelerate this process. They recognize that it's an issue. So this is getting better. But there's a danger, too, to accelerating the process, because sometimes drugs get approved on a provisional basis and then they turn out not to work as well as we anticipated or have more side effects than we anticipated when they're eventually studied in larger trials. So faster approval, often a good thing, but there can be downsides. Much of what we use in oncology is what's called off-label use. So drugs may not be formally approved for a given disease, but we use them because there's compelling evidence that they're active in that disease. And one of the ways to get a payer, whether it's Medicare or a private payer, to cover a drug off-label is if there's a consensus amongst experts that the drug is beneficial. So there's a variety of these guideline committees in oncology, the American Society of Clinical Oncology, the National Cancer Control Network, and others that convene panels of physicians who are considered expert in the field, and they make recommendations on what appropriate treatments may be. And even if those treatments are not FDA approved, if these physicians reach a consensus that they're useful and appropriate in a given disease, payers will often cover them. Doesn't mean that the co-payment is going to be reasonable. I've seen many drugs used in this setting where the payer says, sure, we'll approve it, and your co-payment's $1,500 or $2,000 a month. Well, technically they've approved it, doesn't necessarily mean that you're going to have access to it because it could be prohibitively expensive. But the more data we can gather, and the more data that these panels have to evaluate and determine that a given treatment may be effective, the more successful we are with arguing with payers to cover given treatments, even if they're not FDA approved. Again, very important, particularly in rare diseases where it's hard to do some of these large trials that the FDA often requires. So speaking of which, what are the challenges of clinical trials in rare diseases like Erdheim-Chester? Well, we don't have great preclinical models. I showed you all those fancy mice and petri dishes. We don't have good models in Erdheim-Chester disease, or as many models as we may have in things like breast cancer or lung cancer or some more common diseases. If I'm being honest, sometimes it's difficult generating interest from drug manufacturers. They're for-profit businesses. If they look at, launching a study in an extremely rare disease, if something happens, if there's some unanticipated severe side effect that could derail their entire program in more common diseases. So some manufacturers are hesitant to allow the use of their drugs in rare diseases. I think they're getting better at that, but that can still sometimes be an issue. There's less funding often in rare diseases. We see breast cancer fundraisers all the time, or lung cancer fundraisers all the time. again, to the credit of Kathy and her organization, we're now starting to see some good fundraising in ECD, but it dwarfs in comparison to what we see in some of the more common cancers. There's a limited number of centers with expertise in rare diseases, so there's fewer people initiating these trials. It's more difficult for patients to travel to those centers to participate. And we often say there's more ideas than patients. So as we learn more about the biology of these diseases, it'd be great. We say, great, we should study this. And if you're talking diseases that affect a few hundred people per year and you have 10 different drugs you want to test, there's just not enough patients available to reasonably test those new therapies. So you have to be very selective in which ones you might choose for testing. And then FDA approvals can be challenging in rare diseases only because it can be difficult to conduct larger trials that the FDA often likes to see to grant an approval. Not to end on a down note, despite some of those limitations, I've been very impressed with the success of generating all phases of trials in Erdheim-Chester disease. So I just gave some examples here. None of these are my trials. There are trials looking at long-term outcome of using 2 targeted therapies. So, we talked about it's hard enough to study 1 drug in rare diseases. Here we have a trial of 2 drugs in a rare disease. There's multiple trials looking at that. There's trials looking at the optimal duration of therapy. Do we need to keep people on these treatment for years on end? Can we give them breaks? Can we stop treatment safely at various time points? Dr. Diamond and others have registry trials, so maybe you're not interested in trying a new treatment. That's perfectly legitimate. Maybe you can't travel to one of these centers. Well, there's still ways to participate and still ways to learn. You can participate in these registry data, and it's just simple data collection. How are you feeling? What side effects are you having? What symptoms do you have from the disease? What treatments have you received? And that can still help us learn a lot about treatments and a lot about the disease. And then importantly, quality of life. I'm sure I don't have to tell any of the patients or families in this room that Erdheim-Chester disease affects quality of life, and it's generally the main issue for patients. So, making sure that we're doing everything we can to improve quality of life, whether that's through new treatments, whether that's through other supportive treatments to manage symptoms and side effects from the disease, or what I call ancillary services, so things like physical therapy, nutrition, massage, so other things beyond just medications to help people manage their symptoms and side effects. And people are really taking a strong interest in that. We're starting to generate some important results. So despite some of the limitations I mentioned, I think in this setting in particular there's been tremendous progress, and I've been really impressed by the meeting this year and the types of things that people are doing. So just to wrap it up, because I think I am a little bit over, I apologize. Clinical trials are critical to improving treatments and developing new treatments. They're not for everybody. I fully appreciate that. Participation in clinical trials can be intimidating. I had a fairly minor surgery. I was approached about a trial. For a fairly minor surgery, the consent form is like 32 pages long. So, when I hand those to patients, I'm like, it's no big. Trust me, it's not that big of a deal. And then I had it, I'm like, oh my goodness. And I'm reading the fine print. And, at the end of the day, it wasn't a big deal. But it's when you're on the other end of it, you get an appreciation for what it feels like. It's important to know that we can learn from studies that don't involve treatment. So again, these registry studies or quality of life studies help us tremendously. So those are very important. And as the Global Alliance has shown, there are barriers to conducting clinical trials in rare diseases, but these can be overcome with a small group of committed individuals like we have in this room. And if you're interested in learning more, certainly speak with your doctors and nurses. There are also some resources on the internet. Clinicaltrials.gov is a repository of all clinical trials. You can search by various keywords. So you can search by Erdheim-Chester disease, and it'll tell you what trials may be available. The Global Alliance is very tuned in to what trials are available in ECD. They're an excellent resource, and I would encourage you to continue your interactions with them. So I appreciate your attention, and I'm happy to field any questions.